Hello everyone, I am Dr. Preeti Khulbe and in today's lecture we will be discussing about restriction endonuclease and this is in the subject of pharmaceutical biotechnology second unit. So the in 6th sem it is there. So restriction endonuclease. So when we talk about restriction endonuclease or restriction enzyme or any other enzyme which is related to the biotechnology. So first thing which is there to understand that this restriction endonuclease or restriction enzyme is used for the used in the process of DNA recombination, or we can say recombinant DNA technology, or in the genetic engineering. So how it is used, what is the reason behind using this enzyme, this we will discuss in today's lecture. If you want the lectures related to the first unit, that is related to how the DNA recombination occurs, what is the process of DNA recombination, what are the scopes of biotechnology, so you can also visit the previous videos. Also, this restriction in this unit, the cloning vector is also there. So we have already discussed the cloning vector in our previous video. So yeah. So first of all, learning objective. So upon completion of this topic, the students will be able to learn about the various aspects related to restriction enzyme. What is restriction enzyme? Why it is so important? Where to use restriction enzyme? What is the structure of restriction enzyme? So all the aspects related to restriction enzyme you will came to know after the completion of this unit. Other than that, you will know about the scopes of biotechnology. So, what are the scopes of biotechnology in relation to the restriction enzyme also we'll discuss in this unit. Definition and role of restriction enzyme in the field of pharmacy. So, these are the learning objective. If we talk about the learning outcome, so on successful completion of this unit, students will be able to learn about need of restriction enzyme. Okay, what is the need behind using this restriction enzyme, products using restriction enzyme related to biotechnology. Then next is advantages and disadvantages of restriction enzyme. Should we use restriction enzyme or not, or what type of restriction enzyme we can use, what are those advantages and disadvantages which are related to the restriction enzyme, or which are related to using the restriction enzyme in the process of DNA recombination. So this all thing you will came to know after the completion of this unit. Okay. These are the learning objective and learning outcome which is related to the field of biotechnology. Okay. Next is introduction. So, with the process in biotechnology, it has been required to incorporate the desired DNA sequence with specific activity in the host DNA to modify its function and evolve to sustain with the rapidly changing environment. Okay, so basically we can say that in the process of biotechnology, if we talk about the advancement in biotechnology, so the major advancement or major we can say breakthrough in biotechnology is the recombination of DNA. So for this, it is required to incorporate the specific DNA segment for the specific activity which is related to the specific activity to modify its DNA function. Okay, and it is the very useful thing which is used in the future also. So basically we can say that DNA, one segment from the another DNA is transferred to the another segment, another DNA, to modify the activity or to modify the specific property of the DNA. This is the main progress in the field of biotechnology. technology. So the question was how to cut DNA according to the size of a specific DNA of interest. So that is the thing. If you know that how the DNA recombination process occurs, so you must be aware of that, that to recombinate the DNA we need to cut the specific portion of DNA, okay, where that portion which is responsible for the particular activity which we want to modify. Okay, so we need to cut that piece of DNA and then we need to insert it into the another DNA which we want to modify. Okay, so what is the important thing? Important thing is that how we can cut that specific part of the DNA and the size, that is हमको कैसे पता चले कि हाँ वही specific part है और वहीं से उसको cut करके हमको specific property मिलनी है. Okay, so this problematic question was solved by the introduction of the special endonucleases which produce internal cuts in the DNA molecule. For example, or it is known as restriction endonuclease. Okay, so basically before that it is very difficult to see that specific site, then cut it. So how to cut it is very tedious. But after the evolution of this endonuclease enzyme, we came to know that this enzyme is used to specifically cut the DNA, or it is used to produce the internal cuts in the DNA molecule. Okay, so this restriction enzyme endonuclease or this restriction endonuclease enzyme are basically used to form the internal cuts into the DNA molecule. Now, endonucleases are enzymes that produce internal cuts in nucleic acids. Okay, so basically we know that in DNA nucleic acids are there, or it is a we can say a form of a chain of nucleic acid which is the DNA. So that nucleic acids we can make internal cuts in the nucleic acids by using the endonuclease enzyme, or we can say by using the restriction endonuclease enzyme. Okay, the endonucleases that cleave DNA okay, only within or near those sites which have specific base sequences are known as restriction endonuclease. Okay, now how the restriction endonuclease cut that specific portion? So basically there is a specific base sequence in which this restriction endonuclease enzyme is produce the cut. So that is, we can say these when we add the restriction endonuclease enzyme in the in the DNA mixture, so in the DNA solution, so what they used to do, they used to detect that specific base sequence. There are number of base pairs are there, adenine, guanine, thiamine, cytosine, so they can detect those base pair sequence and then produce the internal cuts into that particular portion. Okay, so this is what the role of restriction endonuclease. So I think now you will understand that restriction endonuclease is an enzyme which is used to produce the internal cuts into the DNA, and by the use of this enzyme we can we can get the specific portion and size of the DNA of interest. Okay, and basically these endonucleases specify or detect that specific base pair sequence, and they cut from there. Okay, now another important thing is nomenclature of restriction endonuclease. So how we can nomenclate the endonuclease? So the first letter of the name of genus in which a given enzyme is discovered is written in a capital. Okay, so that is there are a number of restriction endonucleases enzyme. This is not one enzyme, that restriction endonuclease, but there are number of enzymes which are used as restriction endonucleases. So their name, first letter of the name is from the genus, okay, from which the enzyme is discovered, उसके genus के नाम पे होगा. उस बैक्टीरिया और उस वायरस उसके जीनस के नाम पे there is a first letter, then this is followed by the first two letters of specific name of the organism, that is other two letters are related to the specific name of the organism. Okay, now these three letters are generally written in italics. Okay, so here you can see the example that eco from Escherichia coli, hin from Haemophilus influenza, HAPA from Haemophilus para-influenza. Okay, so here you can see eco, E is from Escherichia, which is the genus, and CO from the species. Cola is the species, so first two letters of the species, so eco. Similarly, here the genus is Haemophilus, so H is there, and influenza, in is there in that hen. Similarly, HPA, so H is HEPA, so H is from the genus, that is Haemophilus, and para-influenza, PA from the para-influenza. Okay, so this is the bestest way to nomenclate the restriction endonuclease. Now, when an organism produces more than one enzyme, okay, so they are identified by sequential Roman numerals. The different enzyme produced by H influenza strain, DR are named hen D second, hen D third, etc. Okay, so here the we can say that here the example is given. So if for example two or three enzymes are produced by the same organism, so हम कैसे करेंगे? जैसे H इन्फ्लुएंजा, इससे ही आपका हो रहा है दो तीन एंजाइम, ई कोलाई उससे दो तीन एंजाइम हो रहे हैं. So generally we used to identify it by the sequential Roman numerals. So basically जैसे जो first evolve हुआ है उसको हम 1 नाम दे देंगे. Here you can see hin H influenza 2, that is this is the second which is evolved in this. Okay, after that कोई अलग आएगा तो उसको 3 नाम दे देंगे. So this is like that. Okay, all restriction enzymes are designated by the journal symbol R, which is prefixed to their names, like RECO R1, R Hindi 3, similarly R BAM H1, etc. Okay, and R, wherever R is there, so R represent the restriction enzyme. Okay, so here prefix R, this is the important thing. कई बार मैं questions में पूछती हूँ कि what is R represent for, तो लोग इस वाले R को समझते हैं, but R prefix is there, the restriction enzyme which is, this is the strain name basically. Here you can see that E Escherichia, CO coli, R is strain name, first is the order of discovery Roman numerals. Okay, so this is, we can say, the very good example of the nomenclature of that. Now, types of restriction endonucleases. So there are multiple types of restriction endonucleases are there. First one is type 1. So type 1 restriction endonucleases have following properties. These have recognition sequence of about 15 BP, that is 15 base pair recognition sequences there. They cleave the DNA about 1000 base pairs all away from 5' and to the sequence. Okay, so that is the important thing, that this restriction, if you want that your properties between in the 15 base pair, between the 15 base pairs, so they can recognize up to 15 base pairs only. Okay, and they can cleave the DNA after the 1000 BP, that is away about 1000 BP tak. So away from 5' end. So 5' end जो है, you know that the DNA structure has 5' and 3' end. So from 5' end 1000 base pairs के बाद it can easily cut the DNA, and 15 base pair tak it can cut that portion. Okay, so the sequence of cutting is non-specific. So it is not specific, it is not non-specific. We'll discuss what is this non-specific and specific sequence in detail further. Now it require S-adenosyl methionine ATP as well as magnesium 2 plus for its optimal activity. Okay, so we can say that here the S-adenosyl methionine, if it is not there, so it cannot produce cut. So basically in the solution where we are taking the DNA and restriction endonuclease type 1 enzyme, so we need to add S-adenosyl methionine ATP as well as magnesium 2 plus for the optimum activity, for its optimum activity, or we can say to produce the cuts. Okay, now the it consists of three subunits. Okay, so this type 1 enzyme which we are talking about, so it consists of three subunits, which is HSDR, which it required for the restriction, HSDM, which is required for the methylation of host DNA, and HSDS is important for specificity of recognition, so DNA binding site. Okay, so basically HSTR is required for the restriction. Okay, so there are three subunits, this you should remember. HSTR, R stands for restriction, M stands for methylation, S stands for specificity of recognition. So basically we can say it can HSD as basically recognize the site where we have to make cut. Okay, M is then do the methylation by using this adenosyl methionine. Then this HSDR जो subunit होगी, it restrict it or it can produce the cut, basically we can say. Okay, so this is all about the type 1 restriction enzyme. The examples are E. coli K, E. coli B. Okay, etc. Now the type 2 restriction enzyme, so these are the simplest and commonly available and use restriction endonucleases. So type 2 is the very commonly used restriction endonucleases for the purpose of DNA recombination. Now these are made up to two separated identical units having the endonucleases and methylases activity. Okay, so as in the type 1 we see that there are three subunits. Similarly here two subunits are there which are identical, but for one is having the endonuclease activity and another one is having the methylase activity. Okay, examples are E. coli R1 and hen D2, etc. So we have already discussed these two names. So what these do, so they cut the DNA at defined positions close to within their recognition sequence. Okay, so where the recognition sequence is there, from there only they can cut the DNA, and they do not use ATP and adenomat, that is adenosine methionine, for their activity, but usually required only Mg2 plus as cofactor for their activity. Okay, so it is very easy to do, because we need to take a solution, or solution in that we need to add just a DNA and type 2 restriction in it, and only magnesium ions are required into it, because Mg2 plus के अलावा इसको और कुछ भी नहीं चाहिए. They do not need ATP as ATP and adenosyl methionine as in case of type 1 it is required. Okay, their sequence of cutting is specific. We have discussed in the type 1 that sequence of cutting is non-specific. So similarly for that the sequence of cutting is specific. They produce discrete recognition fragments. So because it is the specific type of mechanism, so they produce specific recognition fragments also after the cutting. The pieces or the fragments are very specific. Okay, so we have already I have already told you that we'll discuss what is the specific, non-specific. This is very simple and we'll discuss it in detail in detail further. Okay, now the recognition, now the type 3, so restriction endonuclease. So this is the third type of restriction endonuclease. It recognize two separate asymmetric sequence that are inversely oriented. Okay, so yeah, So inversely oriented asymmetric sequence they can easily recognize, and they cut DNA about 20 to 30 base pair after the recognition site. As in first we have discussed that they produce the recognition site के 5' end से 1000 base pair के बाद ये 15 को cut करता है, and second में we have discussed that it cuts close to the recognition sequence. So here the type 3 is which is produce the cuts to the in the DNA about 20 to 30 base pair after the recognition site. So जहाँ पे भी हमको cut चाहिए, basically according to that we can select the type of restriction endonuclease. Okay, now it consists of two subunits. Type 3 is consists of two subunits. Type 1 has 3 subunits. Type 2 also has 2 subunits, and type 3 according to that type 3 is also there, which is having the 2 subunits only. First is the mod. So subunit recognizes the DNA sequence and is a modification methyl transferase. Okay. Similarly, so DNA sequence को recognize करना है. Second, res is subunit which is required for the restriction. They require adenosine, methionine, and ATP cofactor for their role. Okay, in first one, ATP, methionine, adenosine, methionine, and magnesium ions are required. In second, only magnesium Mg2 plus is required. In the third, the adenosine, methionine, as well as ATP cofactors are required. So examples are ECO P1, ECO P15. Okay. So now the fourth one. So restriction enzyme type 4. So type 4 restriction endonuclease modified the typically methylated DNA and are exemplified by the methyl MCR, BC and MRR systems of the E. coli. Okay, so these systems can be exemplified by the use of DNA. Okay. Now type 5. So these enzymes utilizes guide RNAs to target the specific sequences found on organisms. They can cut the DNA of variable length and a suitable guide RNA is provided. Okay, so this type 5 basically RNA utility is there. So it utilizes the RNA guide and target the specific sequence found in the organism, and they produce the internal cuts also. So the cuts are in variable length, and it is again guided by the RNA. Examples are the Cas9G RNA complex from the CRISPRs. Thing. So basically this is a type 5 which is totally guided by the RNA. Okay, so we have discussed that there are 5 types of this basically we can say restriction endonucleases are there. Okay, just revise quickly. Type 1 is recognition sequence is 15 base pair. It cleave the DNA after the 1000 BP from 5' ends. It's required S-adenosyl, methionine, ATP and Mg2 plus cofactor. It has 3 subunits. Second is it has 2 subunits and it can separate, they can meet the 2 separate identical subunits are there, and in this required ATP and adenosyl methionine is not required, only Mg2 plus is required. Similarly in type 3 the recognize 2 separate asymmetric sequence and that are inversely oriented, and they cut DNA about 20 to 30 after the recognition site. It consists of mod and res sites. Type 4 is it also has the thing that the enzyme modified, these are the enzyme modified type of recognition site. Typically it is methylated by DNA and then exemplified by the other systems. Similarly type 5 is totally related to the RNA. Now type 2 restriction endonuclease are widely used in recombinant technology. So as we have discussed also, the type 2 is very important, and it is type 2 restriction enzyme, and it is used in the recombinant technology. So this is due to their ability to catalyze the phosphodiesterase bond and cleavage with very large rate enhancements, while also maintaining execute sequence selectivity. Now restriction site, so it is also known as restriction recognition site. ठीक है, ORI हमने देखा था, origin of site. Similarly it is the recognition site, and restriction, it is also known as restriction site or restriction recognition site, because this is the site from where the restriction in the enzyme is occurs. Okay, now these are locations on the DNA molecules. These are locations on the DNA molecule which contains a specific nucleotide sequence and are recognized by restriction enzymes. Okay, a particular restriction enzyme cuts the sequence between two nucleotides within or near its recognition site. Okay, so basically it is the site of recognition, and these are locations in the DNA molecule and specific where the specific nucleoside sequence is there. Okay, so basically this restriction enzyme recognizes that sequence. Okay, so particular restriction enzyme cut the sequence between two nucleotides within near to the recognition site. ठीक है, जैसे we have discussed also that type 2 is specifically cut near the restriction recognition site. So कोई क्या type 3 is clave or restricted it from the after the 15 to 20 base pair from the recognition site. Okay, so similar to that. Now the cleavage patterns. So most type 2 restriction endonuclease cleave the DNA molecule and produce two types of cuts. Okay, so after the cleavage there are two types of cuts are produced. One is the staggered cuts, and second one is the blunt or even cuts. Okay, so staggered cuts are two DNA strands are cleaved at different locations. Okay, this is known as the staggered cut. When the restriction enzyme attack to the DNA molecule to produce the cut, so cut may be staggered or may be blunt or even cut. Okay, so staggered cuts are basically two DNA strands are cleaved at different locations, and this generates 3' or 5' protruding ends, and which readily pair with each other under aniline conditions. Okay, so these ends are called cohesive or sticky ends. Okay, so this is the important thing, that in staggered cuts the cleave at different portions are there, so these are not even basically we can say, and these can generate the 3' and 5' protruding end, and these are also known as sticky and cohesive end. Second is blunt end. So here you can see some restriction endonucleases cut both strands, the strands of the DNA molecule at the same site, producing blunt and even flush end. Okay, so here you can see that अगर क्या होगा आपका restriction endonuclease same जगह से cut करेगा, so it will produce the from both the ends, so it can produce the specific or even and flush ends. Okay, so now the mode of action. So generally the process consists of the following steps. First one is the recognition of the binding sites. Second is binding of the enzyme dimer to the DNA. Okay, it can first recognize the binding site, then it can bind with the enzyme dimer to the DNA, then cleave the cleavage of the DNA, then enzyme releases. Okay, so these are the four mode of actions in which first the recognition of binding site is necessary. Then after the recognition it can bind with the enzyme dimer of DNA, then it can cleave it, the cleavage of the DNA occurs, then enzyme is released after the cleavage. So here you can see in the first one, you can see this is the structure of DNA, and here the recognition site is there. So first thing is recognition of this site, second is here you can see we have discussed about specific and non-specific binding. Okay, so here two types of main, basically this is the recognition site of DNA and this is the restriction enzyme homodimer. Okay, so here again the binding is occurs in second step, binding occurs. So in binding may be type non-specific or specific. So here you can see the non-specific binding, this is the recognition site and it binds the restriction enzyme here. Okay, so this is the non-specific because it is not bind directly to the here, directly to the recognition site, but in specific the restriction enzymes bind in the direct that site only. Okay, so after that the linear distribution, now coupling here. So here releases, as this is the thing, so it is like this and it is like this, so it means this. The next is the cleavage. After binding, cleavage is their third step. After cleavage again ये क्या क्लेव हो गया, break हो गया, then this enzyme is released, restriction enzyme. After it's all the process, after the whole process अपना सारा काम करने के बाद it can be released and relaxed. Okay, so this is the process, or we can say mode of the restriction binding is there. Okay, so here we have discussed about the type 2 restriction enzyme is basically responsible restriction sites where we have discussed that it can recognize easily, and from there the restriction enzyme act, it is known as restriction site. Cleavage pattern, we have discussed that there are staggered and blunt end. Staggered cuts are two DNA strands are cleaved at different locations and it forms the cohesive or sticky end, that is known as the staggered cut. And secondly the blunt cut, so some restriction endonuclease cut both strands, the strands of the DNA molecules at the same site, producing the blunt, even or flush ends. Okay, so here you can see blunt ends में क्या होगा, equal portions बनेंगे and उसमें नहीं बनेंगे. Mode of action, we have discussed that there are four mechanisms. First is site recognition, second is binding, third is cleavage, and fourth is enzyme release. So here in the chart it is very specific that first step is the recognition, so it can recognize the binding site and this is the restriction enzyme. So in second step the binding is there, so if there may be a non-specific, that is the far from the restriction recognition site, then specific, it is on the recognition site, so it may bind accordingly according to the mechanism. Then there is a cleavage, it can break this enzyme and then the enzyme will be released after the cleavage. Okay, so this is the whole process, this we have discussed. Now the applications. So as we have discussed about a lot related to the restriction endonuclease enzyme, so now I think you will be able to know that what are the applications. So there are many applications of the restriction endonuclease. A few of them are mentioned here, like restriction mapping and inserting foreign gene. Obviously either we want to insert the restriction mapping. So restriction mapping, so for example uh, here we have we have like by breaking it into pieces and then identifying the locations of the breakpoints. This method relies upon the use of proteins called restriction enzymes, which can cut or digest DNA molecules at short, specific sequence. So basically the restriction mapping is there, which is used for the specific recognition of a specific sequence and then cleave it as per the thing, that is the restriction mapping. Similarly inserting foreign genes, so inserting foreign gene is that if we want to insert the specific sequence of gene into the another sequence, so they can make the they can do the they can incorporate the specific DNA sequence there in that particular thing. Okay, so here you can here the these are basically two very basic and very common applications of DNA of this restriction endonuclease. Other than that, other specific mechanism includes the that DNA recombination. In case of DNA recombination, the restriction enzyme is very essential or very useful. Now the conclusion. So the restriction endonucleases are very essential enzyme which are able to cut the host DNA and create a favorable site for the insertion of a DNA segment of interest. Okay, as we have discussed, it is a very important enzyme and it can create the insertion, or it can create the cuts to the to that enzyme. Okay, it produces sticky or blunt end as per their recognition sequence, and thus it is a valuable tool for modern biotechnological studies. So if you want to produce the sticky and or blunt end, we have discussed that staggered and the even or blunt things are there. So if you want to produce that type of different cuts by as per the recognition sequence, so also this restriction enzyme may be used. Thus it is a valuable tool, we can say that too, for the modern biotechnological study. Among of all its classes, class 2 or type 2 is more favorable for restriction mapping because it because of its ability to produce discrete fragments. Okay, because in that discrete fragments are produced, so we can say it is it can be used for this restriction mapping. Specifically type, there are 5 types, but type 2 is the most favorably used or most commonly used. Now restriction endonucleases are indispensable. Restriction endonucleases are indispensable for DNA cloning and sequencing. So if you want to do DNA cloning, as we have discussed also in the last class about the cloning vector. So if you want to do the DNA cloning and sequencing, so they serve as the tool for cutting DNA molecules at predetermined site. Okay, so they can easily, we can say, they can detect the predetermined site, and they can cut they can serve as a tool for the cutting of that DNA molecule. Now, which is the basic requirement for the gene cloning of recombinant DNA technology? So, if you want to do the rDNA technology, if you want to clone the DNA by using rDNA technology, so this is the prime requirement. This is the basic requirement that you should produce the cut to the should produce the internal or cut to the cut into the that specific portion of DNA, and you can then you need to fix it. So this is for them. Okay, so these are some important things related to restriction endonuclease. So we can say that restriction endonuclease is an enzyme which is used to produce the internal cuts to the DNA molecule, and it is a very important thing for the restriction for the recombinant DNA technology. For the restriction mapping, it is very essential. For cloning of DNA, it is again important. Other than that, it has 5 types, in which type 2 is very common. It may produce blunt or sticky ends also. It may produce even ends also. And the mechanism, if we talk, so its mechanism may be it may be like it may be mechanism if we talk, so yeah, it may be specific or non-specific. Also, if we want to talk about the mode of action, which is very very important, it has four steps in mode of action. First one is recognition of the binding, second is binding of the enzyme dimer to the DNA, third one is cleavage of the enzyme, and fourth one is enzyme release. So here we have already discussed all these four steps. Okay, so other than that, we have discussed all these things. The introduction, internal nomenclature, we have discussed that it's genus name. First one is genus name. Second, two letters which are in the small and which is acts as the species name. Okay, yeah. So we have discussed all of things related to that restriction endonuclease. So these are the references. Fundamentals of Medical Biology is a very good book which you can prefer. Also, U Satyanarayan is a very commonly read books by all the students and related to the biotechnology. So you can visit any book, you can read any book. Also, you can directly cite it through the Google and for the detail, and also you can check out the things on the internet for the recent updates related to that. Okay, if you want to go in detail or recent updates you want, so you can visit the other books or other journals also. And if you want to study in detail related to rDNA technology, related to cloning vector, related to biotechnology and its application, so I have already made video for that. You can watch those videos. So yeah, thank you so much. Thanks for watching my video.